What if I told you that extinction isn't forever? Because right now, in the middle of Texas, there are wolves walking the earth that haven't existed for over 10,000 years. Not statues, not museum displays, real, living creatures with ancient DNA coursing through their veins. They look like they came straight out of an Ice Age documentary, and they were created in a lab. This isn't some sci-fi movie pitch. It's the work of a company called Colossal Biosciences. In just a few short years, they've gone from concept to functional de-extinction, meaning animals that aren't just genetically similar to extinct species, but are designed to fill the same role in the wild. It's one of the boldest, most controversial scientific missions ever attempted, and it's already happening. They didn't start with a woolly mammoth or a saber-toothed cat. They started with a predator that once ruled North America during the Ice Age, the dire wolf. Using DNA pulled from two ancient wolf specimens, one over 13,000 years old, and the other a staggering 72,000, they identified and reprogrammed 20 key genes. These genes controlled things like skull size, bone strength, muscle density, and even fur color. Then, they did something that shocked the scientific world. They used CRISPR gene editing to splice those ancient genes into the DNA of modern gray wolves. And from those edited cells, they cloned embryos and implanted them into domestic dogs. The result? 3 living, breathing creatures named Romulus, Remus, and Kessie, born in 2024 and 2025. They may not be exact copies of dire wolves, but they're close enough to raise eyebrows, headlines, and serious questions. Because now that we've brought back one extinct predator, who's next? But hold on, wolves were just the beginning. And what Colossal did next pushes things even further into sci-fi territory. After bringing back wolves with ancient traits, Colossal didn't slow down. Their next move? Something even stranger. Turning regular lab mice into tiny versions of a woolly mammoth. It might sound like a weird science project, but there's a real reason behind it. Before scientists can bring back something massive like a mammoth, they need to test what works, and mice are perfect for that. They're small, they breed fast, and scientists can see results in just weeks instead of years. So here's what they did. Colossal gathered DNA from over 100 mammoths and elephants, some of it more than 1 million years old. They studied the genes that helped mammoths survive the cold, like ones for thick fur, fat storage, and body temperature control. Then they picked the 7 most important genes and used CRISPR to insert them into lab mice. The result? A new breed of woolly mice with golden wavy fur and extra body fat, just like mini mammoths. It might sound funny, but these little creatures are a huge step forward. In less than a year, scientists created 38 of them. And now that they know the gene edits work in living animals, they're getting ready for the real test, mammoths. But before jumping to giant animals, there's one more reason mice matter. Scientists need to be sure that these ancient genes don't cause problems, things like organ failure, immune reactions, or unexpected mutations. If the mice stay healthy, it's a green light to move to the next stage. And that next stage? It's something the world hasn't seen in over 4,000 years. This is where things start to feel unreal. Colossal's ultimate goal isn't just to make furry mice. It's to bring back a creature the world hasn't seen in thousands of years. The woolly mammoth. Not as a museum exhibit or a CGI animation. A real living, breathing mammoth calf. And they're aiming to do it by 2028. Here's how they plan to pull it off. Mammoths and Asian elephants are about 99.6% genetically the same. So instead of building a mammoth completely from scratch, Colossal wants to edit Asian elephant embryos to include dozens of mammoth traits. We're talking about things like thick fur, small ears, a hump of fat for cold survival, and resistance to freezing temperatures. They're planning to change 65 to 85 genes to turn those elephant embryos into mammoth-like hybrids. Once the edits are done, the embryos will be implanted into real living elephants, who will carry the baby mammoth for nearly 2 years. And if all goes well, that calf will be born with the ancient traits of its Ice Age ancestors, ready to walk the earth for the first time in over 4,000 years. But why bring back mammoths in the first place? According to Colossal, it's not just for show. They believe reintroducing mammoths could help restore frozen grasslands in places like Siberia. These animals once helped maintain the ecosystem by knocking down trees, spreading seeds, and packing snow. Scientists think mammoths could even help slow down climate change by keeping permafrost from melting. It's a bold idea, some say too bold. But Colossal is moving fast, and their confidence is real. Because if mice worked, and the genes are ready, then the mammoth might be next. But while Colossal is focused on bringing mammoths to life, scientists in China just did something else that shocked the world. They cloned a living wolf. And this time, it wasn't extinct, just endangered. While Colossal was making headlines with mammoths and wolves, scientists in China pulled off something completely different, but just as mind-blowing. In June 2022, the world met Maya, the first cloned Arctic wolf. She wasn't extinct. Arctic wolves still roam the wild, but their numbers are shrinking fast due to habitat loss and climate change. So scientists at a biotech company in Beijing called Syngene decided to do something radical to help protect the species. They cloned one. Here's how they did it. They took skin cells from a wild Arctic wolf that had died and removed the nucleus, which holds the DNA. Then, they took an egg cell from a regular dog and swapped out its nucleus with the wolf DNA. Once the embryo was created, it was implanted into a beagle dog. Yes, a completely different species, and the beagle carried the pregnancy to full term. The result, Maya, a healthy, living Arctic wolf clone. This was a huge breakthrough, not just for cloning, but for conservation. It showed that endangered animals could be cloned and possibly saved from disappearing forever. No complicated gene editing, no mixing of traits, just a clean copy of a rare animal, born through science. Maya might not be a de-extinct animal like the dire wolf projects, but she represents something just as important, a way to stop species from vanishing before it's too late. And while Maya was making history in China, researchers in Australia were preparing for something even more ambitious, bringing back an animal that's been gone since 1936. And it wasn't even a wolf. It was something stranger. The Tasmanian tiger wasn't actually a tiger. It wasn't even a wolf. It was a strange, striped marsupial, kind of like a cross between a dog and a kangaroo. Officially called the thylacine, it once roamed across Australia and Tasmania. But after years of hunting, disease, and habitat loss, the last known one died in a zoo in 1936. For decades, people thought that was it. Game over. But now, that might be changing. In 2017, a team of Australian scientists decided to try something that had never been done before. They dug into old museum specimens, preserved bones, fur, and tissue, and managed to sequence the thylacine's DNA. It wasn't perfect, but it was enough to start something big. Their plan is bold. Take the DNA of the thylacine's closest living relative, the Tasmanian devil, and slowly rewrite it using CRISPR gene editing. Bit by bit, they're replacing the devil's DNA with thylacine traits, everything from body shape to metabolism. It's like rebuilding the animal from the inside out. But that's not all. The real challenge is how to bring the embryo to life. Scientists are exploring two options: implant it into a surrogate marsupial or grow it in an artificial womb, something straight out of science fiction. It's still early, and no thylacine has been born yet, but the progress is real. And if they pull it off, the thylacine would become the first animal brought back from extinction after being gone for nearly a century. And that raises the question, if we can do this with one animal, where does it stop? Because in North America, scientists are trying to revive a creature that once darkened the skies in flocks so big it looked like a cloud was moving. Next up, the return of the passenger pigeon. There was a time when the skies over North America would go dark. Not from clouds, but from birds. Passenger pigeons once flew in flocks so massive they could stretch for miles and block out the sun. People said you could hear them coming from miles away, but by 1914, they were gone, wiped out by overhunting and massive habitat loss. It's hard to believe that something so common could vanish forever. But now, scientists are trying to reverse that story. The plan? Use DNA from museum specimens of real passenger pigeons and combine it with the genes of their closest living relative, the band-tailed pigeon. The result won't be a perfect replica, but it'll be close. A hybrid bird with many of the same traits as the original passenger pigeon. But it's not just about bringing the bird back for show. These birds played a big role in shaping forests. When they landed in huge flocks, they'd break branches, open up the tree canopy, and drop tons of droppings that actually helped fertilize the soil. So researchers believe that bringing them back could help restore forest ecosystems that haven't been the same since the pigeons disappeared. To test this, Scientists in Michigan ran forest simulations, basically mimicking what a massive flock would do to the environment. The results? More light reached the forest floor, more plants started to grow, and the soil improved. Now, the real birds are next. The team is currently raising band-tailed pigeons that carry CRISPR-edited DNA, meaning those genetic changes can be passed on to their chicks. They're aiming to hatch the first passenger pigeon-like birds by 2029. It's not just about undoing the past. It's about fixing what was lost. But pigeons aren't the only animals being brought back from extinction. In Europe, scientists went even further. They cloned a wild mountain goat that hadn't walked the earth in years. And for a brief moment, it actually worked. In the mountains between Spain and France lived a rare kind of wild goat called the Pyrenean ibex, also known as the bucardo. Strong, tough, and built for rugged cliffs, this animal had survived for centuries, until everything changed. By the year 2000, there was only one left, a female named Celia. Then tragedy hit. Celia was found crushed under a fallen tree. Just like that, the Pyrenean ibex was officially extinct. But scientists weren't ready to give up. Celia's skin had been saved and frozen, and in 2003, researchers used it to try something never done before: bring an extinct animal back to life. They took her DNA, inserted it into egg cells from regular goats, and implanted the embryos into goat surrogates. And then, something incredible happened. One of the embryos developed. A baby ibex was born. It took its first breath, becoming the first extinct animal ever cloned back into existence. But the moment didn't last. The baby had serious lung defects. Just 7 minutes after birth, it died. It was heartbreaking, but also historic. For the first time, science had shown that extinction could be reversed, even if only for a few minutes. The cloning worked. The embryo developed. The ibex lived, however briefly. In that short moment, the impossible became possible, and it pushed other scientists to keep going. Because if one extinct animal could be brought back, maybe others could survive too. One of the most urgent cases, a giant, powerful species that was down to just 2 females, no males, no future, until a team of scientists came up with one last plan: to save the northern white rhino. By 2018, the northern white rhino was nearly gone. There were only 2 left in the entire world, both of them female. That meant no natural way to keep the species going. No males, no hope. At least, that's what people thought. But a team of scientists from Kenya, Germany, and the US weren't ready to let the rhino vanish. They came together with one goal: bring back the northern rhino. Here's how they're doing it. They started by collecting eggs from the 2 surviving females. Since there were no living males left, they turned to frozen sperm, taken years earlier from northern white rhinos that had already died. In the lab, they used in vitro fertilization, IVF, to create healthy embryos. But that's just one part of the challenge. Rhinos have incredibly long pregnancies, about 16 months, and IVF in rhinos is extremely difficult. The embryos also need a mother to carry them. So the plan is to implant these embryos into southern white rhinos, a closely related species, who can act as surrogate moms. If it works, the baby would be a true northern white rhino, just born from a different mother. So far, the scientists have successfully created multiple embryos and frozen them. Now they're working on the final step, getting one of those embryos to grow into a living calf. It's a race against time, but there's real hope. This could be the first time a species that was functionally extinct gets a second chance at life, not through cloning, but through careful, high-tech reproduction. And if it succeeds, it won't just save the rhino, it'll prove that even the rarest animals can still have a future. But this kind of breakthrough didn't start with rhinos. It all began with a sheep named Dolly, the first cloned mammal in history. And she changed science forever. Before wolves, mammoths, and rhinos, there was Dolly, the sheep that changed the world. Back in 1996, scientists in Scotland did something no one had ever done before. They took a single cell from an adult sheep, removed the DNA, and used it to create a brand new baby lamb. That lamb was Dolly, the first mammal ever cloned from an adult cell. And in that moment, cloning moved from science fiction to science fact. Here's why Dolly was such a big deal. Before her, cloning had only been done using cells from embryos, early undeveloped cells. But Dolly proved that even a fully developed adult cell could be reprogrammed to create a brand new life. That discovery changed everything. It opened the door for cloning more complex animals, saving endangered species, and even, one day, bringing extinct ones back. Dolly wasn't just a lab experiment. She lived a normal life, gave birth to 6 lambs, and became a global symbol of what science could do. She lived for 6 years before passing away from a lung disease, but by then, she had already made history, and her legacy lives on in every cloning project that came after. From Maya the Arctic wolf to Romulus the engineered dire wolf. Without Dolly, none of this would be possible. But while cloning started in the lab, it's now being used for something even more important, bringing real animals back into the wild, not just keeping them in test tubes or zoos. And the next animal on that list? A tiny predator that was once nearly gone, now getting a second chance. Let's talk about the black-footed ferret. In the early 1980s, the black-footed ferret was thought to be completely extinct. These small, secretive animals had once lived all across the Great Plains in North America, hunting prairie dogs and keeping ecosystems in balance. But by then, they had vanished from the wild. Then came a miracle. In 1981, a small group of black-footed ferrets was discovered in Wyoming. Conservationists quickly moved to protect them, starting a breeding program to save the species. It worked, but there was a problem. The ferret population came from just a few animals, which meant they were all genetically very similar. And when genes are too alike, it can lead to health problems, lower survival rates, and fewer chances of long-term survival in the wild. That's when scientists turned to cloning, again. In 2020, a group called Revive and Restore used frozen cells from a wild ferret that had died over 30 years earlier. From that DNA, they created a healthy baby ferret named Elizabeth Ann. She was born using a regular domestic ferret as a surrogate, and she was the first cloned black-footed ferret in history. But more importantly, Elizabeth Ann wasn't just healthy. She carried rare genes that hadn't been seen in the population for decades. And by adding her into the breeding program, scientists are now helping increase genetic diversity and give the species a better shot at surviving in the wild. It's not just about cloning for curiosity anymore. It's about using science to protect real animals, real habitats, and real futures. And the black-footed ferret is proof that cloning can go beyond the lab and actually help bring life back into the wild. But there's one more story you need to hear. It's about a wild horse that once roamed the open plains, disappeared, and came back, thanks to a piece of frozen tissue that had been stored for 40 years. This is the incredible case of the Przewalski's horse. The Przewalski's horse, pronounced Przewalski, isn't just any horse. It's the last truly wild horse species on Earth. That means it was never tamed or bred by humans. For thousands of years, these strong, stocky horses roamed the grassy steppes of Central Asia. But by the late 1960s, they were almost completely gone. Overhunting, fences, farms, and shrinking habitats pushed them to the edge. The only ones left were in zoos and breeding centers, a handful of animals, far from the wild. That's where cloning came in. In 2020, scientists used a tiny sample of tissue that had been frozen for over 40 years. It came from a stallion named Kaporovic, who had unique genes not found in the current horse population. Using those frozen cells, they cloned a healthy baby horse named Kurt, born in Texas. A few years later, they did it again, cloning Kurt's genetic twin named Ollie. These clones brought something extremely valuable to the species: fresh genetic diversity. With such a small population, every bit of variation matters. New genes mean stronger, healthier horses that are more likely to survive and thrive in the wild. And that's exactly what's happening. Today, Kurt and Ollie are part of a growing effort to reintroduce Przewalski's horses back into their natural habitat. They're not just science experiments, they're part of the future of their species. This might be the most successful case of de-extinction so far, a wild species, saved by science, and now galloping toward a second chance. And if we can bring back a wild horse from a frozen sample, clone a wolf from skin cells, and build mammoth traits into mice, what's next? That's a question we all need to think about. So, what happens when extinction is no longer forever? From ancient wolves reborn in Texas, to woolly mice in labs, to a cloned horse running wild again, science is rewriting the rules of life and death. What used to be gone forever might now just be on pause. It's not just about bringing back animals for curiosity or headlines. These projects are changing how we think about conservation, climate, and even the future of our planet. Cloning and gene editing are no longer far-off ideas. They're tools being used right now to save species, fix ecosystems, and maybe even reverse some of the damage we've done. But there's also a bigger question. Just because we can bring these animals back, should we? What are the risks? What happens if they don't fit into the world as it is today? And who decides which species get a second chance? One thing's clear. Science has opened the door, and we're already walking through it. The age of extinction may be ending, but what comes next is up to us.